In this video, we will understand how this KRAS downregulates FAS, which is the dead cell receptors on the pancreatic cancer cells. In the previous video, we have understood how the KRAS is negatively or inversely proportional to the T cells activations and directly proportional to the myeloid cells infiltrations. So they want to know, the researchers want to know in depth why the KRAS has that kind of relationship with the T cells and the myeloid cells. So they, in order to dig more into the mechanism, what they did is they developed a cell lines which is doxycycline derived PIKP 785 cell lines. That is, when the doxycycline is introduced, the KRAS mutations are on, and when the doxycycline is not introduced, KRAS mutations are off. And they have also observed that when the KRAS mutations are off, the kinetics of this pancreatic cell lines is slightly slower compared to the case where KRAS mutations are on. And these results are shown in the supplemental figures. And in the validations whether this system is working or not is shown in the panel B, where we can see that when the KRAS mutations are on, they see the high KRAS expressions, whereas when the KRAS mutations are off, after 3 days and 5 days they see a negligible relative expressions. And then they check the single cell RNA-seq data of these different kind of cells, and they have find out that each populations behave differently on the UMAP. It can be seen there. On cell populations, KRAS mutations on populations segregated separately, compared to the case where KRAS mutations are on and off, and the KRAS mutations are off by 3 days and by 5 days. This indicates that the KRAS has a role towards this KRAS has a role towards regulating the gene expressions on the pancreatic cancer cells, and the expressions and the violent plots also shows that the KRAS mutations are expressed differently in the case where KRAS mutations are on and the KRAS mutations are off, and when the KRAS mutations are on again after turning it off, or on after or we can say the on-off conditions of this pancreatic cancer cells. And thereafter they did the GSE analysis, that is gene set enrichment analysis, and the final conclusions they find out is that when the KRAS mutations are off, the T cell proliferations and the T cells activations are upregulated by around 1.5% approximately, compared to the case when the KRAS mutations are on. And after validating that there is a positive impact on the T cell proliferations, then they check the qPCR results to find out what are the genes that are dysregulated in both the conditions. And what they have find out is that when the KRAS mutations are off, the FAS expression levels are highly upregulated compared to the control case, that is when the KRAS mutations are on or KRAS mutations are on, off, and on conditions. And these things are also shown in the this panel G, where we can see that the fold chain expressions of the KRAS mutation FAS receptors. The panel F is about FAS receptors when the KRAS mutations are on and off, and when the KRAS mutations are off. And the panel G is about this some detailed analysis which are detailed results or data which shows that which of the genes are upregulated in the case when KRAS mutations are off by 3 days and 5 days. We can see that FAS is one of the highly upregulated genes that can be seen in these results. And these things they have validated using the this quantitative analysis, where we can see that relative FAS expressions are high when the KRAS mutations are off for a longer period of time. And they also checked using the flow cytometer that at what time this peak of this FAS receptors has been observed, and they find out that after 2 weeks there is a peak in the FAS receptors observed, and this peak will continue if the KRAS mutations if the KRAS is not expressed till 4 weeks. After that, when the KRAS mutation KRAS mutations are expressed using doxycycline treatment, they see the reduction in the FAS receptors. And these things further validated using the in the panel J, where it shows about the multiple replicants analysis, and it shows that after 14 days it reaches its peak, and which continue till 4 weeks. After that, if they starts the KRAS expressions using doxycycline treatment, the FAS receptors goes down. And after validating that these FAS receptors, dead cell receptors goes up, they wanted to check whether these dead cell receptors cause the death of the pancreatic cancer cells or not. So they use the anti-FAS antibodies and check the apoptosis marker, and they have find out is that when the KRAS mutations are on, the increase in the apoptosis is higher compared to the in a case where FAS antibodies are used compared to the case where FAS antibodies are not used. But when the KRAS mutations are off, they see a significant increase in the conditions where FAS antibodies are used compared to the case where FAS antibodies are not used. And apart from this apoptosis of the cells, they also check another apoptotic marker, that is caspase-8 absorbance, and they see that caspase-8 absorbance also increase in the case where KRAS mutations are off and the FAS antibodies are used. So from these results, they made a point that KRAS mutations has a negative role towards FAS receptors expressions on the cancer cell surface. And if the KRAS mutation goes down, there is a increase in the FAS receptor expressions, and that can interact with the FAS ligand that are expressed on the immune cells, and that can trigger apoptosis of the tumor cells. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.